Hey guys, so today we're going to talk about chapter 1. So chapter 1 is a very generalized overview of like the history of microbiology and some basic things that differentiate what is a microbe. So let's get started. So, what is a microbe? A microbe is generally defined as a living organism that requires a microscope to be seen. So, generally speaking, if you can see it with your naked eye, it is not considered a microbe. But there are some, of course, exceptions to every rule. So there are some like protists, algae you can see with the naked eye, such as like an amoeba. Also, another exception will be with like fungi, so like fungus. They form something that's called a biofilm, and a biofilm is a multicellular structure, and there are different cell types within that structure a lot of times. So an example is that fungi, they form these biofilms and you can actually physically see it, and you can see it on like mold on a bread. A lot of times that can be considered a biofilm. Also, a biofilm on pretty much any surface, so you can see that. And then as well as there are some parasites, which is like roundworms and whatnot. You can also see that with the naked eye. But generally speaking, a microbe requires a microscope to be seen. So, microbes and how they are important to us. So, microbes, they actually generate air that we breathe, so they do produce oxygen, carbon dioxide, and nitrogen. They can also they also produce essential vitamins that we need. So vitamins that we aren't able to make ourselves, we have to actually consume. So we can, such as like vitamin B12, vitamin K. Also, there is a human relationship with microbes, such as like through food production, food preservation, as well as some biotechnology tools. We'll talk about more the biotech side of it later on in the semester. Then also medically. So medically, we use microbes in order to produce something such as like vaccines. We use them a lot, primarily, like I said, with vaccinations. So how do we name our microbes? So there was a scientist. He came up with a way of creating their names. So whenever you see a microbe's name written, you'll see it in two names. So the first name is a capitalized name of their genus, and then the second name is the lowercase version of their species. So such as you see below, Staphylococcus epidermis. So that is how it would be written, and that's actually how I expect you to write it too later on in the semester whenever we write some papers. You can also abbreviate it, such as the one below, as you see E. coli. You can abbreviate them, and the way that you abbreviate it is that you take the first letter of its genus, capitalize it, and then you write out their species name. Okay? So, with microbes, they are distinguished into three different domains. So, the three different domains of life is what our whole world is made up of is bacteria, archaea, and eukarya. And then all three of these domains, we all come from a common ancestor. So somewhere along the lines, there was something like one organism, and then it eventually diverged. It evolved into something completely different, but we all stem from the same thing. Okay? So, like I said, there are three domains of life. We have bacteria, archaea, and eukaryote. And then also within those three domains, we have another subsection called kingdoms. So, such as like Animalia, um, Plantae, so like the plants. So Animalia would be us, like mammals. Also some worms, but mainly just mammals for the most part. And then, like I said, like Plantae and everything, those are all part of the eukarya, as well as fungi and protists. Those are all part of the eukarya domain. So just know that there are the big picture, three domains of life. Within the three domains of life, there are multiple kingdoms, and then so on. Okay, So now we're going to talk about what distinguishes, like what is the big thing among these domains and kingdoms. So bacteria is prokaryotic. So meaning prokaryotic means that generally they're really small, like you normally can't see them with the naked eye. Also, it means that they don't have any membrane bound organelles, such as like a nucleus. So, like with our cells, we have membrane bound organelles such as like a Golgi apparatus, um, an endoplasmic reticulum. Nucleus is the big one. So, we'll talk more about those organelles later on. But bacteria, know that they are prokaryotic. They are pretty small. They are generally no more than 20 micrometers. Also, they generally are single celled and they grow within communities. So, like, they'll be individual little cells, but they are together collectively. And then also, they don't have sexual reproduction. They do something that's called asexual reproduction, specifically binary fusion. So essentially, they copy themselves. And then also, bacteria, they have cell walls, whereas human cells, we do not have a cell wall. So those are the big things that distinguish a bacteria. So now, archaic cells, they're also prokaryotic, but they are genetically different than bacteria. And generally, archaea species, they live in extreme environments, like I said, inside of like volcanoes or the excessive cold, like in the Arctic ice. So since they live in such harsh environments, they're known as extremophils. And then also they're non-pathogenic, meaning that they don't cause any disease. Whereas bacteria can cause disease. Archaeas, they are non-pathogenic, so they don't cause any disease. Now, protists are eukaryotic, so they're more like us. They are those single-celled organisms, but they are modal, so they are free living. So they can live outside of something, so they don't have to live with inside another animal. And they're generally considered parasitic. Now, algae are also eukaryotic, but the difference is that they contain chloroplasts. So since they contain chloroplasts, kind of like a plant cell, they they do photosynthesis. And then also they have cellulose within their cell wall, which helps them with their rigidity, with their shape. Okay, now fungi, my favorite. So fungi, they're eukaryotic, but they're non-modal. So they cannot um, they don't have any appendages that help them move. Like whereas like as we saw as with the protozoas, they're kind of like wiggly, so they're kind of like worms, like slime. So they're able to move. They are modal as well as algae. Algae are considered modal to a degree, but fungi, they're non-modal. So most of y'all probably think fungi, you think of a mushroom, which is true. But fungi, they're heterotrophic, so meaning that they have to consume their own food, kind of like us, where they're not autotrophic, meaning that they can produce their own food. So fungi are heterotrophic. They grow either as a single cell or hyphae. So hyphae, we'll talk more about hyphae later. It's a different cell type where they are kind of elongated cells and they are connected to each other, but they are still individual cells. But like I said, we'll talk about those later. Fungi, they are known to cause disease, and fungi, they do have cell wall, but they do not have cellulose. They have something that's called chitin in their cell wall. So that's another big thing that distinguishes fungi from all others. Fungi is the only one that has chitin in its cell wall. Okay. So another microbe are viruses. So viruses are non-cellular, so they are technically, in my opinion, not alive, but they do carry genetic material. So genetic material is either DNA or RNA. We'll talk more about the differences between them later on. But what viruses do is that, kind of like the flu virus, what will happen is, or the HIV virus, you'll be exposed to the virus and it'll take over our cells. And then by taking over our cells, it produces more of itself. Viruses are pathogenic, so they do cause disease, like I said, like the flu, HIV, HPV. But with viruses, we do produce vaccines with them. That's why we have a flu vaccine, an HPV vaccine. So viruses are very important, but they are considered non-cellular. Okay, so now we're going to transition into the history of microbiology. So over the years, microbiology has definitely evolved and it has played a huge role in our history. So throughout history, actually, like soldiers would die because of these infections that would happen, and they didn't know what was going on. So with like studying, essentially studying the dead, studying our history, we were able to develop the medicine and the microbiology practices that we do have now. And we're going to talk a little bit about them. And then also, we learned how to use microbes to our advantage, such as creating vaccines, creating food and drink. So, definitely, the history of microbiology is essential. So, we're going to talk about some scientists that played a big role. So, one of the biggest ones was Robert Hooke. He was the first one to build the compound microscope, and he's actually the first one to publish a picture that he saw while using his microscope. And he's the one who actually coined the term cell. So the reason why he called them cells is because he saw them as like individual little units. And then actually there's a picture on the slide that shows you what he saw. That's actually uh, his drawing that he saw while using the microscope. And he primarily used mites and nematodes to observe this. Another scientist was Anton. He observed bacteria with a single lens microscope, so a different kind of microscope. He was the first one to actually successfully observe a bacteria. But even though he was able to observe a bacteria, um, in history, bacteria or like microbes were not linked to diseases yet. So with Robert Hooke, he had a weaker microscope, but then Anton, he created lenses that were stronger than the ones Hooke was using. Okay, so we're gonna transition into some theories. So spontaneous generation. So spontaneous generation, it is the theory that was created by, I'm gonna butcher his name, but it was Spalzellini. I think that's how you say it. So this guy right here, his name. So he's the one who came up with the theory that living microbes can arise spontaneously without any parental organisms, meaning that you don't have anything, then all of a sudden, poof, it's there. So what his experiment, what he did is essentially the first image that you see. So he had some gravy, so that was kind of like the growth medium, like the food. What he did was that he boiled it and everything, so to essentially clean the gravy, and then he left the flask open. And then with the flask being open, he saw that there was some microbes in there. Then he did it again, but instead of leaving the flask open, he had it closed. And then with the closed flask of gravy, it showed that there was no microorganisms. But with that, there was some people who argued that since the flask was sealed, oxygen was not readily available. So the scientist who argued him the most was Louis Pasteur. So what he did is that he created a swan neck flask, which is the picture on the bottom. So this swan neck, this S shaped. So with this, he he said, okay, you think that we think that microbes need oxygen, so let's create this S snake, this S shape thing. And with that, they were they were allowing oxygen in, but also keeping the growth medium free of any contamination. So with the swan neck flask, Pasteur actually disproved spontaneous generation. So Louis Pasteur was the one who disproved spontaneous generation with the S neck. Okay, so now we're going to talk about Florence Nightingale. So Florence Nightingale, she was the first one to actually publish a textbook on nursing. So she was the one who linked infectious disease to mortality. So she came up with something that's called germ theory, meaning that a specific disease is caused by a specific microbe. So meaning that if you get exposed to the flu virus isn't going to cause meningitis. It's only going to cause flu. So, like I said, Florence Nightingale, she's the one who showed the significance of mortality due to disease. So essentially, if you had a very bad infection or some kind of ailment, a lot of times your mortality rate was very high. So unfortunately, you were going to die, at least in this time period. So, with that linkage between disease and death, somebody named Kosh, he came up with an idea of how to actually physically link a pathogen to his disease. So he came up with something that's called Kosh's postulates. So there are four postulates. So the four postulates is that a microbe has to be found in all cases. So meaning, let's use flu as an example since it's very easy. So the flu virus, let's say that we have 10 patients, they all exhibit flu. So everybody has to have the flu, and then we have 10 patients that are healthy. So from those 10, from the 10 who have the flu, they all have to have flu. They can't be caused by something else. So then, unfortunately, after those patients die, the microbe has to be able to be isolated and then grown. So we have to be able to grow up the flu. And then what we need to do is that once we have just the flu microbe, what we do is that we infect our healthy host. And then once the healthy host is infected, the healthy host will get sick and will die from the disease. From that, after the new host die from the disease, you have to be able to obtain the microbe again. So those are the four Kosh's postulates. So as you can see in this image right here. Okay. So Kosh, he was the first one to be able to establish that there was a link between a microbe and an actual infection. So he was the first one to actually truly get a solid link between microbe and disease. So with that, sort of segued into immunizations. So immunizations have actually been around for a while, been around for thousands of years. So immunizations, the idea between getting immunized is to prevent a disease. So it's in order to completely avoid you getting sick. So the first marked of where immunizations was being used was actually in India and China. So it was known that individuals could become immune with smallpox by transferring secretions from an ill person to a healthy person. So, what you did is that you essentially controlled the person's exposure to smallpox. And then later on in the 18th century, the occurrence of smallpox was decreased by actually purposely inoculating children with smallpox. So that was the first idea of vaccines, I guess. So by prophylactically inoculating a child with smallpox is actually very dangerous. And some individuals actually still got sick from it. But although it did dramatically decrease, so we'll actually talk more into how this was further evolved into a safer version. So, what we have today is a very safe version of inoculating us with diseases. It's a very safe way. Whereas back then, they didn't know the things that we know, but it was still very good. And then one of the first people to ever do this was Dr. Edward Jenner. So he would actually deliberately infect patients with cowpox. And then with that, it helped with immune response, and it also decreased mortality rates. So that was the idea of a vaccine. Edward Jenner was the first one to coin the vaccination term. So we talked a little about Louis Pasteur before, but we're going to go more into detail about him. So Louis Pasteur, he was the first one to show that infecting somebody with a weakened version of it was able to boost immunity. And he also applied this principle to different organisms, so not just bacteria. So Louis Pasteur, he came up with the idea of, okay, vaccines, like vaccinating is a good idea, but there's still increased sickness. So how can we make it better? So how we make it better is we weaken the microbe. So we make our body the stronger host as compared to the bacteria or the fungus or whatever organism it may be, which is what we actually do today. So we now know that there are certain, they're called molecular components of a pathogen. So pathogens have multiple different little parts to them, and we know that certain parts of that actually generate an immune response by our immune system. So we do know that. We'll talk about that later on in the semester when we have our discussion about vaccines. But what Pasteur essentially did was he studied cholera, and he isolated the bacteria. And what he did was that he left out that bacteria in heat, essentially, and then with the excessive heat, it weakened that cholera bacteria. And then with actually inoculating somebody with the weakened bacteria, that cholera, it actually failed to cause disease, but yet you still had the immunity to it. And he tested this in chickens. So now we're going to transition into antiseptic and some old school antibiotics. So antiseptic and antibiotics, the big idea behind them is that they want to control the pathogens. So long before Pasteur and Koch, a lot of people died because of the lack of antiseptic techniques. So the first doctor to ever order somebody to wash their hands with chlorine actually was I'm not even going to attempt to say his name, but it was this gentleman. So he actually ordered his doctors to wash their hands with chlorine. And chlorine is actually a known antiseptic and it does kill microbes. And with him ordering his doctors to wash their hands, mortality rates dramatically fell. Like they fell. So there was less deaths essentially. And then the next person to also enforce antiseptic techniques was Lister. So he saw that a lot of amputee patients died from sepsis, which is an infection of the blood. So what he did is that he started using a different antiseptic agent. He used carbonic acid in order to treat the wounds and the surgical instruments. So that also helped as well because with acid it is very acidic. So it does kill microbes as well. And then also it kills the wounds. But as you know, you don't want to put acid on yourself because we know that it does eat away flesh. So that's not good, but it definitely did decrease the amount of infection. So that gave the patients a better chance of living. So even though with antiseptic advances, they the patient still would die, unfortunately, because antiseptics can only go so far because they're only, for the most part, they're all external, whereas a lot of times you're fighting an internal battle. So that is whenever research went towards antibiotics, which was considered the magic bullet of the time. So, antibiotics, what it does is that it actually just kills the microbe and it doesn't hurt the host. Whereas antiseptics, a lot of times, like as we saw with Lister, he used acid that can be very harmful to the patient. Whereas antibiotics are specifically targeted to a microbe and it leaves the patient unharmed. So ideally, you want to use antibiotic treatment rather than just antiseptic treatment. But definitely, you need a clean environment in order to prevent infection as well. So that's why we always say wash your hands and everything. That's why doctors, they always scrub up before and everything. So we definitely need antiseptic techniques, but not so harsh antiseptic techniques where it was hurting our patients. Okay, so another scientist was Alexander Fleming. He was actually he was the one who found the mold that killed bacteria. So it's the penicillium mold. So Alexander Fleming, what happened was he was culturing a staphylococcus bacteria and his culture got contaminated with mold. But upon viewing his culture, he saw that where the mold was growing, there was no bacteria growing around it. So, with that, he saw that the mold was secreting something that was saying that was killing off bacteria or not allowing bacteria to grow. So, as you see in the picture, so we have our mold, which is that giant white dot, and then we have like this kind of clearing area around the mold. So this is where bacteria was not allowed to grow. So he was just like, huh, I wonder what's going on. And then with some experiments and everything, penicillin was discovered. So penicillin was discovered by accident, guys. Okay, so now we're going to transition into the discovery of viruses. So the discovery of viruses happened in, I believe, in around the 1890s. So it was done by a scientist named Dimitri. So he observed that there was an infection going on plants, and he saw whenever he tried to culture what was going on with the diseased plants, he saw that the porcelain filter that he was using retained the bacteria. So he saw that the disease was the causative agent of the disease was still passing through this filter. And with that, about 5 or 6 years later, another scientist made the exact same observation, which was Martanius. So Martanius, he saw the exact same observation. So he saw that the porcelain filter was still holding the bacteria, but the disease causing agent was still passing through. So that concluded that the pathogen must have been something completely different and it wasn't a bacteria. And then as as time progresses, there was somebody named Wendell Stanley. He is the one who observed um, the infectious particle, and he coined it the tobacco mosaic virus. So he's the one who actually, in a way, purified the causative agent. So what the way it started was that Dimitri, he saw that there was something causing a disease in the tobacco plants, and he made the observation that bacteria was being caught inside the filter, but the actual causative agent was passing through this filter. And then Martanius, he made the exact same observation, so he concluded that the pathogen is completely different. It's completely different than a bacteria. And then later on, Wendell Stanley, he crystallized that infectious particle. And he's the one who said, okay, we're going to call it the tobacco mosaic virus. So that's a very basic overview of how viruses were discovered. We'll talk more about different viruses later on in the semester. I believe it's chapter 12. So that is just the basic idea of how a virus was discovered. Okay, so one of the last concepts that we're going to talk about is something called endosymbiosis. So endosymbiosis is whenever a living organism lives symbiotically, so meaning it's not causing any harm, it's just there, inside a larger organism. And endosymbiosis is widespread throughout multiple ecosystems. And the person who came up with the theory of endosymbiosis was Lynn Margulies. I believe that's how you pronounce her name. So her theory proposed that eukaryotic cells were able to develop these complex compartments like a mitochondria and chloroplast because way back in the early days, what happened is that one cell uptook something that actually had mitochondria and chloroplast, and then they merged into one cell. So here is a figure that shows what I mean. So we have a pre-eukaryotic cell, so that means like pre-mitochondria, pre-chloroplast, everything. So pre all organelles essentially. So we have this prekaryotic cell, and then we have a bacteria cell. So what happened is that a bacteria cell that had like mitochondria or had chloroplast in it got uptook by this prokaryotic cell. I'm sorry, pre-eukaryotic cell. So we had bacteria that had chloroplast in it and a bacterium that had mitochondria in it. And then we had this prekaryotic cell that uptook either one of these, and then it evolved into either plant and algae, into animal, fungi, protists, or just it stayed a bacteria, either way. So the idea is that there was a bacteria cell that had chloroplast and mitochondria, and then there's another cell that didn't have either of them, and then the cell uptook and they merged into one thing. And then that is how we got chloroplast and mitochondria into our cells. So that is the idea of endosymbiosis. Okay? And then also the reason why endosymbiosis is a theory is because mitochondria and chloroplasts actually have their own genetic sequences. So that's another reason why endosymbiosis is a favored theory. Okay, so now we're going to go into the history of DNA. So there so the history of DNA, the structure was actually discovered by using something that's called X-ray crystallography. So this was a method that was developed in Europe in the early 1900s. And the two scientists who discovered the structure of DNA was Dorothy Hodgkins and Rosalind Franklin. So these are the two women that used X-ray crystallography in order to discover the structure of DNA. And the image actually at the bottom is an X-ray crystallography of DNA. So X-ray crystallography was used to discover DNA structure, and that was done by Dorothy and Rosalind. Okay. So two other scientists that were pioneers in the, I guess, sort of discovery of DNA was James Watson and then Francis Crick. So there's a lot of controversy actually behind who discovered DNA. Um, but generally speaking, the two people who discovered DNA, the structure of DNA, was Dorothy and Rosalind. And then James and Francis, what they actually did, they didn't do any physical experiments, meaning what they did was that they used other people's experiments and from everybody else's discoveries they compiled their knowledge. So, but from everybody else's knowledge, Watson and Crick, they were able to determine that DNA had certain base pairs, and from those base pairs they had to be paired correctly. So like you can't put a circle through a square hole. It has to be a circle and a circle hole. So that was from all their knowledge, that's kind of what they discovered. And then also they actually got a Nobel Prize for their discovery of the structure of DNA as a double helix. But it unfortunately, Rosalind was not able to be acknowledged in the Nobel Prize because she died. So, as a Nobel Prize, in order to get one, you actually have to be living. So that's the controversies behind it. Is that Watson and Crick, they used her knowledge. They used Franklin's knowledge without. They used her knowledge without her consent, and they got a Nobel Prize out of it. Okay, so that was our first lecture, guys. So we talked about what is a microbe. We talked about how they got their scientific name, so like their genus and species. We talked about the three domains of life, and then also we talked about the differences between the microbes, like prokaryotic and eukaryotic. So different, like what actually distinguishes between bacteria, archaea, protists, fungi, and so on. We talked about various scientists that shaped microbiology. We talked about cautious postulates, the theory of endosymbiosis, and how the structure of DNA was discovered. So, if y'all have any questions, please don't hesitate to email me. So, hope y'all enjoyed our first lecture.